Today we're going to talk about nervous system, and this is going to be at least 2 lectures. We're going to divide this in half, and then we will have a system on sensory. That will be our last lecture before our next test. So as we talk about the nervous system, this is one of the control centers of the human body. And I say one of the control centers because you also have a chemical control system that's called the endocrine system. So both the chemical and these electrical signalers are important in controlling everything we think, say, and do in the human body. So from the nervous system standpoint, we can divide this into a set of cells and signalers that are going to bring information into our nervous system, and these are going to be your senses. We're going to talk about your 5 special senses and some generalized senses that basically gives your nervous system information about the state of the environment and the state of your internal environment, where your body is in space and how your body is functioning. So we bring in information, but then your central nervous system also is going to take that information and basically crunch it together much like a computer would to make sense of it. It's called integration. So we're getting information about our body in space, about gravity, about the temperature, about our blood pressure, about how much fluid volume we have in our body. So all of this information gets all put together to give us an idea of how our body is functioning. And it does this extremely rapidly. Milliseconds, not seconds-wise, milliseconds is the rate at which this information moves. And so for our discussion of nervous system, we're going to divide the nervous system into 2 major categories. The first major category is going to be called the central nervous system, or the CNS for short. And the CNS is going to contain the brain and the spinal cord. But connected to the brain and the spinal cord are all of our nerves that go out to the peripheral parts of our body. And therefore, we refer to these as the peripheral nervous system. And notice our PNS is divided into 2 categories, and this is dependent upon the direction of information transfer. The sensory division is information that is going to the brain. It's coming into our central nervous system. That information coming in from the sensory division gets integrated in the central nervous system. So think of the CNS as the integration center, the computer for the body. And then that information leaves the central nervous system and it goes out to a target. That division is called the motor division. So in the sensory, we can sense what's going on. We're cold. The brain integrates that information. We need to warm the body because our core body temperature is falling, because our atmospheric temperature is low. The motor information goes out to your muscles to cause you to shiver to try to warm the body. So do you see the sensing, the integration, and then lastly the action that results from the motor division? So again, what we're doing here is we're looking at the direction of signal or information transport. Sensory is going to the brain. Motor is going away from the brain or the central nervous system. Now we're going to break this down a little bit more. And again, when we start these units, we're talking about big, broad overviews. Like I said, we like starting simple and add detail as we move into the chapter. So in our peripheral nervous system, we can take the motor division, the division where information is leaving the brain, and we can further divide it into 2 subdivisions. One is called the somatic motor system, and soma means body. So the somatic division is going to be those motor nerves that are controlling the movement and the action of skeletal muscles. Versus the autonomic division, which is going to control our other two types of muscle as well as the secretion of glands. Now, this term autonomic, does it sound like another word that you use frequently? How about automatic? If something is automatic, do you have to think about it? No. So the autonomic division, this is going to be the division that is under involuntary control. Go ahead and write that in there. We're going to expand on that as we get to those separate divisions. But go ahead and write in for autonomic. This is involuntary control. Whereas skeletal muscle is going to be largely voluntary in nature. Now, do you remember we already talked about a skeletal muscle that can be used subconsciously? You remember that muscle? What was it? Tried to slam it, it wouldn't slam. The diaphragm. Do you remember with breathing? You don't have to think about it. But you can control it. You can stop it. Now, when we get to this last autonomic division here, understand too, we can further subdivide that. So we took our nervous system, divided it into central and peripheral. We took peripheral, divided it into sensory and motor. We've divided motor into somatic and autonomic. Now we're dividing autonomic into two divisions. Make sure you get that outlined just right so you can follow the flow. But this division that we have involuntary control over can be divided into sympathetic and parasympathetic. Again, we're going to get into more detail, but here on this slide, for sympathetic, I want you to write in your margin, sympathetic fight or flight. Fight or flight. That division is going to be the involuntary control that gets your body extremely active to do something. Either run away from something that's trying to get you or run down something that you really want. Now, in many instances, when you look at biology and you have divisions in two, when you learn the definition or the function of one, the other is always going to be the opposite. So if sympathetic is extremely active, what do you think parasympathetic is going to be? None. So sympathetic is fight or flight. Write this down for parasympathetic. Rest and digest. Rest and digest. When you're sleeping, that's when you're primarily going to be digesting what you eat. And this is why you don't want to eat right before you go to sleep, because everything you eat is going to get stored as fat. So if you can go for about 3 hours before you go to sleep without eating, your body's going to be in good shape, not to just store fat when you sleep. Do you remember this word, antagonistically? We talked about the biceps and the triceps working in opposite directions. These work in opposite directions, get you active, put you to sleep. And the state of whichever one is most active is going to control what you do. During the day, sympathetic is largely in control. When you're asleep, parasympathetic is largely in control. And so it's going to be a tug of war on these all day long. Now, has anyone ever experienced the sympathetic division kick in like that? Just instantaneously? Have you ever been sitting at a red light? Your light turned green and you start to pull out. Somebody runs the red light right in front of you and it almost hits the front of your car. What do you usually do to the steering wheel? White knuckle? What happens to your heart rate? Your heart starts beating out of your chest. There's usually a few words that jump out of your mouth that you probably don't want your mom to hear, right? That's sympathetic division. How long does it take for that to get kicked into high gear? instantaneously. That sort of gets you to the speed with which all of this stuff happens. Once again, I'm a picture person, right? So here is our overall view of the nervous system. Central is going to be our upper blue oval. There you see brain and spinal cord, information going to the brain, leaving the brain. Peripheral nervous system is our lower box with the sensory information on the left in yellow and the motor division on the right in green. Somatic is the darker green upper division. And then here is our lower purple autonomic division divided into parasympathetic and sympathetic. Does that sort of help you understand these, where they belong and where they fit? Again, when you're studying for the test, use pictures, use abbreviations, use mnemonics, use songs or rhymes, whatever it takes to help you remember the larger, more important information. Don't just try to memorize all the words and all the text on every slide. Okay, that's going to bog you down a lot. So does everyone understand the big global view of nervous system? All right. We started with the big overview. Now we're going to get into the components. So we're going down to the cellular level now. And the signalers within our nervous system, whether we're talking peripheral or central, are the cells that transmit electrical signals, and these are our neurons. But the odd thing about these signalers, which are kind of like the stars of the movie that you watch on the TV or the big screen, neurons are only about 20% of the cells of the entire nervous system. When you watch a movie, most of the people responsible for the production and the working of that movie aren't on screen. And so with our nervous system, we're going to have 80% of the cells of the nervous system supporting the main stars of the show, the neurons. Well, the stars of the show are going to have essentially three main components to them. There's going to be a cell body, which we call the soma. And there are going to be these two types of processes that extend away from this cell body, making them look like a star with all these points. And some of these processes are kind of small, and you have quite a few of them that basically receive information and bring it toward the cell body. Those processes are called the dendrites. Now, I don't have a good mnemonic for dendrites and remembering that it brings information to the cell body. I used to say dendrites bring information down to the cell body because it starts with a D. But in fact, the other process is a little bit easier. And that other process is called an axon. And there's usually only one of these per neuron. It's going to be larger in diameter. But the axon sends information away from the cell body. See, that's easy. Axon starts with an A, away starts with an A. And if you remember axons away, then remember dendrites are the opposite, meaning bringing information down to the cell body. So again, main cell body, two different types of processes, the dendrites and the single axon. Here is like a kindergarten illustration, but I like it because it's simple. Science doesn't have to be complex. You see our cell body over on the left, it's got a nice nucleus illustrated. Many of these small processes radiating away from the cell body, these are in fact the dendrites. And so which direction is information traveling through a dendrite? To or away from the cell body? To the cell body. We have this one singular, much larger diameter process, much longer process, the axon. This is where information is going down away from this cell body to the target. It may be another neuron, it may be a muscle, or it may be a gland. But the axon delivers the signal to the target, whereas dendrites bring the information down to the cell body of the neuron. So I want to look at types of neurons, and we're going to stick sort of with peripheral here, because this kind of relates to how we can have a reflex. Have y'all seen the knee-jerk reflex at the doctor? They tap you with the hammer and your leg kind of jerks. Well, these neurons are involved in that process, so that's why we're going to start with them. So in the peripheral nervous system, we have neurons that are bringing information in, and so we call them sensory neurons because they're going to be part of the sensory division of our nervous system. And these are going to receive various stimuli: pressure, temperature, pain receptors. We have plenty of those. And so these come into and they form connections inside the spinal cord. And in the spinal cord, these sensory neurons very often will form connections with cells that are only inside the spinal cord. So now we talk about central nervous system. And these are called interneurons. Inter because they're between the neurons. And typically we're going to see interneurons between the sensory and our motor neurons. So let's say we touch a hot stove. We'll sort of talk about a reflex already. Have you ever done that? A hot pan. The cookie sheet is what always gets me. I don't know why. I'll bake the cookies or bake something else and I'll forget it's hot, grab it. But as soon as you touch that hot surface, what happens? Woo! You move your hand away before you even know what happened. Because the sensory information coming in, that's hot, that signal gets rapidly transferred through an interneuron directly to the motor neuron of your arm. So that you're not even consciously aware of what's going on. And the motor neuron then transmits that information to your muscles, your skeletal muscles, to cause your hand to move. Now, at the same time, we have other interneurons that go up to your brain to say, that was hot, you're stupid. But do you see the difference between those? Here we have, in this case, we get a splinter, pain receptors activated in our skin in this illustration. Here we have our axon coming all the way down. Once we get into the spinal cord, that's the box at the bottom, the central nervous system. There's our interneuron that's taking the signal, transferring the signal to a motor neuron that goes out to our muscle that usually makes you jerk your hand away when you feel the pain. Now, you don't necessarily have to have the interneuron. Some reflexes have a sensory neuron that sends a signal directly to the motor and out. Makes it even faster. But in this case, with this interneuron, we can also diversify the signal and send it to different places, like to our brain or to the other side of our body. We have some reflexes that we can look at like that. Now, this then is kind of what I was talking about with that interneuron and diversifying the signal. We're looking at a cross-section of our spinal cord, and we're going to get into the components and the divisions of our spinal cord in a little bit in the chapter. But here we see the black arrows, especially on the left. That's our direction of information transfer. And so if we have an arrow and information coming in this direction and going to the central nervous system, is that going to be sensory or is that going to be motor? It's going to the central nervous system. You remember? Sensory. What's going on out here? We touched something hot. Or in this case, we stepped on a Lego in the middle of the night. If you've not done that yet, when you're a parent, you will. Or whatever sharp toy they're selling to kids then. So, you step on a sharp kid toy in the middle of the night, usually about 2 o'clock. Information comes in. There's an interneuron. That interneuron, in addition to sending signal to the motor neuron and telling you to get the weight off your foot, that information is going up to your brain. You stepped on something sharp, but do you see how other interneurons transfer the signal to the opposite side of the body? All of these purple here in the middle, these are interneurons. and the information goes over to the opposite side and it goes down as well. Why would that be important? Why would you need information to go to the opposite side of the body as well? Think about it. You're walking in the dark. You step on something sharp. What is this reflex going to do for this leg? Tell you to get your weight off that leg. What happens if you get your weight off that leg? You fall down, right? But this is saying get weight off your leg, but at the same time, what is it telling your other leg? Get ready to support the weight. So do you see how it's important for that information not to just stay in one place all the time, depending upon this information coming in? It may have to go all over the body. And so that's a great thing about interneurons, that you can spread or diverge that tissue or those signals to other parts of the body, especially to other sides at times. So neurons, sensory, inter, or motor. Those are our three types. S, I, or N. Again, do those abbreviations and make your brain remember the big names of these things. Now, what was the percent of neurons that made up the nervous system? 20, real small. The other 80% of the cells in our nervous system are referred to collectively as neuroglia, or neuroglial cells. And this name neuroglia literally means nerve glue. The cells that support the neurons and all that they do. If you don't have these neuroglial cells, your nervous system is not going to work. And so in our illustration, what we've got is, you see this little purple cable right here? There's another purple cable, our purple. The purple are the neurons. But all of these other cells, the orange ones, the little red one down here, the green ones, those are all neuroglial cells that are supporting, nursing, cleaning, assisting our neurons in their ability to actually send a signal. So one of the first types of neuroglial cells, and we're not necessarily going to cover them all, but are the cells that insulate axons. And we're going to have two types of cells that do it depending on where the neurons are. We have a certain type of cell in the peripheral nervous system, and those are called the Schwann cells. Dr. Schwann is the one that, you know how scientists always stick their name on stuff? Guess who discovered the Golgi apparatus? Dr. Golgi. Guess who discovered the mitochondria? No, that's not Dr. Mitochondria. I don't know what his name was. Anyway, Dr. Schwann discovered these cells. Now, have you ever done electrical work or seen somebody working with electrical wires? Putting in a light switch, a socket, a ceiling fan. If you have two wires that you're twisting together, they have these cool things now called wire nuts. I love wire nuts. But if you don't have wire nuts, what do you have to put around it? This black roll of what? Electrical tape. Electrical tape. Now they have some shrink wrap, and that's cool stuff too. Back in the day, we just had this big black roll of electrical tape. And you'd see electricians, and my dad did a lot of this. My dad was terrified of electricity. I don't like it either. But my dad would wrap a little bit around those bare wires, and then he would put his finger in the hole and just go bzzz. And I mean, he would put a big old knot, almost as big as your hand. Or I guess he didn't want the electricity to leak out. I don't know. But what is he doing? He's insulating those wires so that that signal, the electrical signal, can't short from one place to another. Well, these Schwann cells will contact an axon, and they will wrap their plasma membrane around and around and around and around that axon maybe 50 times, so that you have this layer upon layer upon layer of phospholipid, much like electrical tape. All the cytoplasm gets squeezed and it stays on the outside. It's just phospholipid, this waterproof insulation around it. And when you look at those 50 layers of phospholipid, histologists has given that the name of myelin. And this whole covering that goes around the axon they call the myelin sheath. Now, when we get to the stage where we look at the actual electrical signal and see how the signal goes down the axon, listen to this closely. Your axon, your nerves, can send a signal at a meter per second. A meter per second. That's pretty good for cells, right? But when you wrap myelin around those axons, it speeds it up. Now, a meter per second, that's not going to set a world record in a 100-meter dash, is it? Is the record under 10 seconds now for 100 meters? It's not Usain Bolt. Usain Bolt lost the world record, I think. Someone else has it. So a meter per second, that's not all that impressive. But what we see now when we put our myelin around it, guess what it speeds up to? Would 10 meters per second, would that be impressive? We're getting close to world record there. 100 meters per second. Just because we insulated it. It's crazy. But that's how you can touch a hot stove. And you can move your hand so quickly because of these insulated axons and how fast those signals move throughout your body. Simply because we insulated those layers. Now, in our picture, we see one cell wrapping around one space. Well, this axon may be a couple of meters in length, and we're going to have these little spaces that are going to be between this place what's myelinated, another place. Let's look back at this picture. You see these little segments right there, those three? Each one of those is a myelin sheath, and you see this little space between? Those spaces where you have unmyelinated axons are called the nodes of Ranvier. The electrical signal is basically going to hop from node to node to node to node. We used to give it this big fancy name called saltatory conduction. We have a better understanding of how it happens, but it's still like it goes from node to node to node, and that's what speeds it up over 100-fold the rate in which we can transfer the signal. Oh, I had a picture right here. Duh. So here, do you see these Schwann cells? And one Schwann cell will insulate one area of an axon, but you see all these Schwann cells? And all these little spaces between? The little purple arrows on top are trying to show you how the electrical signal goes from node to node to node to node. And it essentially just basically negates the space that's insulated. So it essentially shrinks the distance with which the signal has to go 100% or 100-fold. So we've already said that our myelin sheath, and the second one here, it speeds up the signal. But the other thing the myelin sheath does, and we're not going to get into a ton of detail here, but the myelin sheath saves energy. Because the way our signal is going to be transported, it's going to be by having gradients of sodium and potassium, these electrically charged ions. And these ions can pass across the plasma membrane through channels. And when they do, after we've sent the signal, we have these pumps that have to pump the sodium back out and the potassium back in, and they're going to require energy to do that. Well, if we've insulated, if we have a watertight layer, and those ions can't diffuse across the membrane, then we don't have to pump them back. And if we don't have to pump them back, we save a ton of energy. So the myelination of axons speeds up the impulse. It saves energy. And in the peripheral nervous system down here on the bottom, when you damage a neuron, when you have a cut in a neuron, if that tunnel of myelinated Schwann cells, if that stays in place, then your neurons can actually use that to guide them back to their target so that they can reconnect in the right place. So in some instances, when you damage a peripheral part of your body, it can be reattached and nerves can regrow. But typically, if you damage the central nervous system, you're out of luck. Because Schwann cells are only in the peripheral nervous system. Make sure you note that somewhere in your notes. Schwann cells are only in the peripheral nervous system. They're only around the axons of your sensory and motor neurons. Now, does that mean that axons in the central nervous system aren't myelinated? No, doesn't mean that at all. We just have different cells that myelinate axons in the CNS. And those cells are called oligodendrocytes. That is a mouthful, isn't it? Oligo means several. Dendro or dendrite means processes, and cyte means cells. So literally, this is a cell that has several processes. And to me, an oligodendrocyte looks like an octopus. You've got the body of the octopus where you have the cell body. And then we have these processes that extend out, and these processes are able to myelinate segments of several different axons. But these cells will only do so in the central nervous system. Schwann cells in the peripheral, one cell myelinates one segment. Oligodendrocytes, however, in the central nervous system, one cell can myelinate several segments of several different neurons. But notice all the layers, kind of like the layers of an onion. The myelin that's present in the central nervous system around those axons, it's just the same wrapping of the plasma membrane of the oligodendrocyte. Just like in the peripheral nervous system, the Schwann cell plasma membrane was wrapping around the axon of the peripheral nervous system. All right, do we get the difference between Schwann cell and oligodendrocyte, and do we understand what myelination is? Think of myelination as insulation. And the cell types basically know which one's in the peripheral and which one's in the central nervous system. All right, we're going to shift gears just a little bit. We started looking at the broad overview of our nervous system. We looked at the cells. Now we're going to talk about the physiology. How is the signal sent? And so the first thing that we need to understand about cells, and again, we're still working with cells here, is that cells are going to have their lipid bilayer, their phospholipid bilayer that you see in our illustration. We've got our polar heads, or the little orange balls, and our hydrophobic tails, or this yellow layer between. The inside of the cell is on the top, and the outside of the cell is down here in blue. Our cells are going to basically cause different ions to be in place either on inside or outside of the cell. And these different ions are going to have different charges. And the cell is going to use energy to actively maintain a certain level of charge around that plasma membrane. And you see in our illustration, you have all these negative charges on the inside and all these positive charges on the outside. Have you ever seen anything that's got a positive end and a negative end? A battery. And a battery has a voltage. And you can get stuff done with a battery, much like the remote I'm holding in my hand or your remote at the house. What happens if you get the battery in upside down? No bueno, right? Doesn't work. So we have to have the charges in the right place on either side of our plasma membrane for the cell to function properly. Now, highlight what you see in the bottom here. The inside of the cell, when the cell's doing nothing, when the cell is at rest, that cell is going to be negative on the inside. And if I go ahead and write this number down, minus 70. We're not going to put units beside it yet. Minus 70. That's the number I want you to stick in your head. When a cell is at rest, the charge across the membrane is minus 70. We're going to add to it a little bit later. All right? So think of the inside of the cell as the negative end of the battery. The outside of the cell is the positive end of the battery. The battery in this case is the plasma membrane. All right. Now the way that this charge difference is maintained is by an active transport pump. We talked about this in Unit 1, right? The sodium-potassium pump. It's going to use ATP, and it's going to transport 3 sodium ions out. That's going to be part of the reason that we're more positive on the outside. And when it resets, it's going to return 2 potassium ions to the inside. Now you go, yeah, but that's positive too. Where are all the negatives? Well, the negatives are already in there. We have so many negative ions that are fixed, they can't get out. That's the main reason that we're so negative inside already. So here's our illustration. Here's our sodium-potassium pump on the left, just this little circle. You can see we have this big yellow arrow or golden arrow going out. It's big because there's 3 sodium that are pumped out. And then when we reset, we bring in 2 potassium to the inside of the cell. Now we're also, for this signaling, we're going to be using 2 proteins that are going to allow ions to pass through them. Now, these are going to be facilitated diffusion proteins. Do you remember that term from Unit 1? Facilitated diffusion simply means these ions will not pass through the phospholipid bilayer on their own, but they will pass through the interior of these protein channels. But these protein channels that allow these ions to pass through are gated. They're not always open. And the key that is going to open these channels is going to be a certain voltage. So write this down somewhere. If you have your slides, you can write them around here or in your notes. These are said to be voltage gated channels. We have a sodium voltage gated channel and we have a potassium voltage gated channel. They are going to open at different voltages. And when we're at rest, and what is our number at rest that I just gave you? Negative 70. These are both closed. At negative 70, both of our voltage-gated channels are closed. We're not sending a signal. Does everyone understand the concept of a voltage-gated channel? It's like a light switch. No voltage, we're closed. Light's off. Flip the switch, electricity, you get voltage, light comes on. In our case, when we get to the right voltage, they will open. And when they open, because of a concentration gradient, that's going to allow the ions to diffuse rather quickly. Because you can think of the plasma membrane like a hydroelectric dam, and think of our voltage-gated channels as the floodgates. When they open the floodgates at the dam, and they might have to do that with all the rain we've been having, when they open the floodgates, is it going to take water very long to start pouring out? No. Because you've got so much water on the side of the lake, hardly any water on the side of the river, the water just pours down because of gravity. Well, the same thing's going to happen with our sodium. When we open that channel, there's so much sodium outside, whoosh, it floods in. And when the potassium channel opens, there's a ton of potassium inside, whoosh, it floods out. And by allowing these charged particles to move either in or out, that's going to change our overall voltage of our battery that we call the membrane of the cell. And that voltage change is going to be the signal. It's kind of complicated, but we're going to put it together. We're just putting the pieces in place right now. Okay? So stick with me. So the first thing we're going to talk about is at minus 70, when the cell is doing nothing. That is called resting potential. Now, typically in my other classes, we also put in membrane, RMP, resting membrane potential. If you need that membrane word to help you understand, then put it in. But the resting potential is minus 70. That's when our neuron is doing nothing. And we're talking about our neurons here in this illustration, because neuroglial cells don't send these electrical signals, just the neurons. So minus 70 is our resting membrane potential. Our units for that minus 70 is millivolt, a little m and a big V, but that's not important. Just stick the number in there. That's what I want you to focus on right now. So at rest, sodium-potassium pump is just kind of idling along. You know, you just started your car and it's just kind of idling. Sodium-potassium pump is just idling enough so that we can keep enough sodium outside and enough potassium inside to stay at our minus 70 number. And the minus sign in front of that 70 lets you know that the inside of the cell is more negative than the outside. If that was positive 70, what would that tell you about the inside of the cell? It's more positive than the outside. But minus 70, the inside is more negative than the outside. And so when we're doing nothing, that is resting membrane potential. That's where we're starting. Now, we're about to send a signal. And this whole ability to send a signal basically from your brain to your big toe, this change of voltage, is called an action potential. Do you see it across the top? Action means movement. Action is, as the name says, active. And so when we change that voltage across the membranes, we are going to create, instead of a resting potential of minus 70, we're going to change and we're going to call these graded potentials. They can be transient. Do you understand what transient means? Temporary, not permanent changes. And they're going to be local, certain spots of our axon or on the cell body, where we change the membrane potential. Either we're going to make it more positive than minus 70, or we're going to make it more negative than minus 70. If it stays at minus 70, we just got a resting potential, right? So as we look in this illustration, it's a little graph. These are our millivolts on the left. This is just time as we click along here. There's our minus 70 millivolts, our green line. You see how it doesn't change? That neuron is resting. It's not receiving a signal. It's not receiving a stimulus. It's not sending a signal. But then all of a sudden we see this first arrow. And when we see that first arrow, do you see how our voltage changes and it goes a little bit more positive, maybe up to minus 60? But what happens after that? It goes right back down to minus 70, back to resting potential. The second stimulus we have, what happens to it? It gets more negative. So all of these changes that are just temporary, that are just local, that happen and then go back to resting potential, those are our graded potentials. You do not send a signal with graded potentials. I mean, you can even have two very close together. Do you see these now just past 60 milliseconds, sorry. You have one, and then before it can reset, you have another. So it's like they're climbing on the shoulders of each other. Because now both of these together are bigger than either one would have been on their own. But still, even with these two together, that's not a strong enough graded potential to send the signal, and so it resets. Now, when I say it resets, I'm talking about the potential. But do you know what is responsible for causing the membrane potential to go back to minus 70 each time? What's transporting the sodium and transporting the potassium? The sodium-potassium pump. Whenever you have these changes, it starts to work a little harder to make sure we can reset just in case we're going to get some more signals anytime soon. So, a more positive graded potential is often called a depolarizing potential. I don't really like that term. I would much rather use a different term here. But we're going to stick with depolarizing at this point. If we make a graded potential that's more negative than minus 70, that is called a hyperpolarizing graded potential. And we're going to see each one of these can have an impact on whether we send a signal or we don't. All right. It's kind of, are we okay here? Graded potential, just making these changes to minus 70, either up or down. Now we're going to put it together into our action potential. This is going to be the thing that zips down our axon. And I want you to think of it like this. Have you ever been in a stadium or a coliseum where the crowd started doing the wave? Somebody stands up, and then the person sitting by them stands up, and that thing just sweeps all the way around the coliseum. How do you know when to stand up and when to sit down? When you're involved in doing the wave, how do you know when to stand up and when to sit down? The closer it gets to you, when the person beside you stands up, you know you get ready. When they start to sit down, you know it's your time to stand up. And then, so your response is dictated by what's beside you. Well, in the axon, many of our voltage-gated channels are aligned side by side, and they influence each other to open. Then the next one knows to open. Then the next one knows to open. Do you see how that's going to be a wave? Except for our action potential, it's going to be a wave of voltage changes. So I want you to look at this illustration and we'll get into the pieces. This is the same sort of graphic, but we've added a little bit to it on the right. Do you see now we have three arrows, three stimuli very close together? And they have added up until they together have changed the membrane potential to a point, and we're going to call that dotted line right there, we're going to call that minus 55 millivolts. Write that number down. I'm only going to want you to remember two numbers. Minus 70 is rest, but I'm also going to ask you to remember minus 55. Because minus 55 millivolts, that dotted line in our illustration, that is the threshold that you see illustrated to start an action potential, to start the wave at the football game. And once you start the wave, it will not stop until it gets to the end. If you start an action potential, it will go all the way to the end of the axon. It will not stop. But you see in our other illustrations, the first one, the depolarization, did we get an action potential? No, because we didn't get to 55. Hyperpolarizing? No, we got even further away, so certainly we didn't. Two stimuli got close, but still not close enough. It took these three. If you have one big stimulus that changes the potential to minus 55, that's enough, and boom, you see that big spike. We have a part going up and a part coming back down. That huge wave, that is our action potential. So at minus 55, we start our action potential. And that upward spike where our membrane potential is getting so positive, that is called depolarization. And this is depolarization during our action potential. Because at minus 55 millivolts, write this down somewhere, your sodium voltage gated channels open. All those positive sodium on the outside comes flooding into the cell. It makes the inside of the cell more positive. Do you see why our spike goes up into the positive numbers on our graph? And it goes up really quickly because of how much sodium we had outside. Well, sitting right beside that sodium voltage gated channel is a potassium voltage gated channel. And when our membrane potential changes to a positive number, guess what the potassium voltage gated channel does? It opens up. And it lets potassium start to flood out, which makes our membrane potential become more negative. Do you see how it's coming back down in our illustration? And the coming back down, because potassium is going out, is called repolarization. So that's the left side, depolarization, the left side of our wave, repolarization, the right side of our wave. And then we will eventually, because the sodium-potassium pump is always working, during an action potential, it goes into overdrive to make sure when we finish an action potential, it resets so that we can do it again very, very quickly. And so what we see written out here in text about depolarization, repolarization, and resetting, we see here in our illustration. So we have our graphic along the bottom, minus 70 resting potential. They don't really show one of our little bumpy stimuli here, but you see how we get up to minus 55. Depolarization, sodium channels open. Repolarization, the sodium channels closed, but the potassium channels open. That's the critical part. And then the sodium-potassium pump is what resets us back to resting membrane potential. Above our graphic, you can see our channels as they're illustrating which ones open and which ones close to give you this movement so that we can get our depolarization with the sodium channel, repolarization with the potassium channel, and then our sodium-potassium pumps are happening during our resetting period. Now, we can only have so many action potentials so rapidly. Because as our sodium voltage gated channels and potassium voltage gated channels are opening and closing, they go through an inactivated period where they're not open, they're not closed, but ions can't move. And it's a brief period of time while they're resetting. And so we call this a refractory period. And that refractory period is absolutely critical to make sure that our action potential only goes in one direction. Think about the football game. If someone decides to start the wave, why doesn't it go in both directions? Have you ever seen a wave go in both directions? No, it always goes in one direction. Well, our action potential only goes in one direction because our channels are resetting that just did the action potential. They can't do it again. And so the action potential will never go backwards. And that's what keeps the signal going from sensory neurons to the central nervous system, and the motor neurons sending it from the central nervous system out to the target. But it's only ever going to be transferred in one direction. So this electrical information running along the axon is like electricity running through a copper wire. But when we get to the end of our axon, all of a sudden the wire stops. And we're going to have a space, a gap. And somehow we have to get the signal from one cell to the next. And that signal may go from one neuron to another neuron. It may go from a neuron to a muscle or a neuron to a gland. But that transfer of information is going to be called a synapse. Or in your textbook, it's called a synaptic transmission. And essentially what's happening in this signaling process, Your neuron is sending electrical information. But when we get to this physical space, that electrical signal is transduced, which means it changes its form. And it's changed into a chemical signal. And we release chemicals into this space that then are able to diffuse from the neuron to the target cell. And that target cell with its receptors picks up those signals. It detects them. And then those receptors in that cells, once they detect the chemicals, that causes a new electrical signal to be generated inside the cell, just the same as it was in the neuron. So think of a synapse, the synaptic transfer of information, being an electrical signal that is changed into a chemical signal that is changed back into electrical signal, only so we can cross this physical space. Because the ions in the electrical signal, they can't jump across the physical space. They have to be in a continuous wire. So this electrical to electrical is called a synapse. And the chemicals then that transmit the signal from the neuron to the target cell, those chemicals are referred to as neurotransmitters. And there are a number of different types of these neurotransmitters. And it depends on what the neuron is, where the neuron is located, or what the cell is that is receiving the signal. All right, so here's our picture. So on the left, here's our axon, and you see our action potential, the arrow coming down the axon. What type of signal is the electrical? I just gave it to you. What kind of signal is the action potential? Electrical. And when we get to the very end, you see our axon spreads out. And all these little green dots that you see inside, those are our neurotransmitters. And they're released in this physical space, this gap. And in this case, our neurotransmitter, the little green ball over on the right, as it's released and it diffuses across this small space, it binds to our receptors. And in many cases, like our skeletal muscle, whenever that neurotransmitter binds, that protein receptor that it's bound to, it may be actually an ion channel. And it causes it to open, and sodium pours in. Doesn't that sound like what started the action potential in the axon? Sodium pouring in? So by the chemical binding, you're causing the ions to change place, and it repropagates the action potential in the new cell. So we've gone from an electrical signal to a chemical signal with our neurotransmitters. Now we've changed it because of our receptors back into that electrical signal. And this may be another neuron, and it's going to send the signal on down the line, or this may be a muscle, in which case that electrical signal spreads across the surface of the muscle cell, causing the muscle to contract. Whether it's skeletal muscle, cardiac muscle, or smooth muscle. So as we're getting toward the end of class now, I want to sort of take a little bit more look at our peripheral nervous system and maybe even get into spinal cord a little bit. But we've looked at the overall sort of characterization and divisions of our nervous system, the cells, and how the signals are sent. Well, let's look a little bit more at our nerves. Now understand, a nerve can be a bundle of nerve cells, a bundle of axons. And we have 12 pair of nerves that come off directly from the brain, but most of our peripheral nerves are going to be spinal, meaning they come off of our spinal cord. And so looking at the spinal cord and thinking about those nerves, we have a part that is going to be dorsal for the spinal cord, and we have a part that's going to be ventral. Do we understand dorsal and ventral? Have y'all covered those in lab before? I like to remember these because when you see a dolphin or a shark in the water, what are you looking for? The fin. What fin is that? The dorsal fin. It's on their back. So your back is dorsal. Your sort of abdomen or your stomach, that's ventral. So if we're thinking on the back or the top of the spinal cord, that is going to be our dorsal root, these branches coming out of the top of our spinal cord. Whereas the ventral root are going to be branches that are coming out of the bottom. Now the dorsal root basically are our sensory axons coming in. Whereas our ventral roots are our motor axons going out of the spinal cord. You see how we're setting up a circuit now for our reflexes? We illustrated those before, coming in from the top with our sensory and going out at the bottom with our motor. Now we're just adding these terms, dorsal and ventral root. So when we talk about reflexes, sensory coming in, motor going out, understand for right now we're talking about a somatic reflex, skeletal muscle reflexes. We're going to get into the sympathetic, parasympathetic reflexes a little bit later. But right now we're talking about the knee-jerk reflex, the touching the hot stove reflex, the tapping the hammer on the knee reflex, and these are all spinal. And this is going to involve a sensory neuron, it's going to involve a motor neuron, and it may or may not involve that interneuron. This is the typical reflex that we see, especially associated with the spinal cord. Now, we keep sort of hammering this in, but as I repeat stuff, you understand that's important to remember, right? So sensory input coming into the spinal cord. Now we're adding the detail that is coming in through the dorsal side. Interneurons are all in the middle of our spinal cord. But the motor neurons sending information away from the spinal cord, that's happening on the ventral part, through the ventral root of our spinal cord. So all those details are important. And this is a spinal reflex, a somatic reflex happening with skeletal muscle. Our autonomic reflexes, sympathetic and parasympathetic, that's all going to be cardiac muscle, smooth muscle, and glands. Somatic here is specifically with our skeletal muscles. And again, this looks exactly like what we saw before, except now we see stepping on the Lego on the left. Information coming in. You see how we're coming into the dorsal? This is the back of your body. So we're coming in through the dorsal root. That's going to be sensory information. Interneurons all on the inside. Here's the ventral root that's bringing that motor information out on the same side of the body to lift your leg so that you're not damaging your foot. But then on the other side, we also have motor information going out that's telling you, okay, contract your muscles to further support your weight. So for this one especially, I want you to just add to the concepts dorsal, sensory, ventral, motor. But again, still the same reflex, still the same thing that we've been talking about for most of our time today. Now, you remember I said you may or may not need an interneuron? Well, in your knee-jerk reflex, when you hit the patellar tendon, that's going to stretch receptors in the tendon. And basically what the doctor is simulating here is you adding a heavy weight to your body. Ladies, I don't think y'all were as stupid as little boys in elementary school, but did you ever, like at recess or whatever, be walking along and your buddy sneak up and jump on your back, just as a joke? And you start to fall, right, because of all the weight, but then many times you don't fall because you catch yourself. You don't think about it, you just catch yourself. Well, that tap of the tendon is simulating somebody jumping on your back. But if your leg is dangling down, the reflex then that's coming in through the dorsal that says heavy weight on our back, and you see that sensory connects right with a motor neuron coming out the ventral surface to your quadriceps muscle, your quadriceps contracts, and this muscle is going to cause your leg to extend. Well, if it's just dangling, when you get that tap, that's why it kicks out there. Because it's simulating that heavy weight, and you've got nothing with your foot dangling, nothing to support the weight, so your leg just kicks out. That's letting the doctor know that your peripheral nerves and your spinal cord are intact. We don't have any problem going on with those areas.